At the beginning of the 20th century, the world's population numbered 1.65 billion people. In 2020, it is predicted to reach the 8 billion mark. This explosion in growth was made possible by something that came to be called the Second Agricultural Revolution, the introduction of specialized plant breeds, and more importantly, the widespread use of chemical nitrogen fertilizers. To sustain this, 1 to 2% of the world's entire yearly energy production goes into the Haber-Bosch process. This energy-intensive process fixes 85 million tons of atmospheric nitrogen for agricultural fertilizers every year. Both of its inventors were awarded with a Nobel Prize. It was termed one of the most significant inventions in the history of mankind. But what if I told you that in the water of our oceans and the soil of our fields, tiny bacteria fix more than twice that amount of nitrogen every year? Their unique properties have been exploited by farmers for centuries and studied by scientists for decades. One of the scientists that dedicated his career to these fascinating microorganisms is Hans-Martin Fischer, professor at the Institute of Microbiology at the ETH Zurich. Professor Fischer, thank you very much for taking time to talk to us today. So, you are working with nitrogen-fixing plant symbiotic bacteria. Can you maybe first explain what these are, what the term means?
Yes. So we are working with bacteria that can live normally in the soil. They are collectively called rhizobia. And as a speciality, they can undergo an intimate interaction with some plants belonging all to one family of legumes. It's a very large plant family that comprises almost 20,000 different species. And the interesting thing about this interaction is that first, these bacteria, when they become the intracellular symbiont of these plants, they can perform a very specialized chemical reaction, and this is the conversion of atmospheric nitrogen into a form of nitrogen which is usable by the host plant.
So maybe our listeners don't know what constitutes a legume. Can you maybe name some industrially or agriculturally important plants that belong to the family of legumes?
Of course, the most agronomically most significant legume crop is definitely soybean. Another crop belonging to the legumes, which is also quite important, is alfalfa, which is a food stock for cattle, for example. And then other species would be beans, which are very important in South America, and peas, and of course also peanuts. These are all species belonging to the Dutch family of legumes.
Yeah, living. Can you maybe tell us more about what exactly it is you're looking into?
Yes, one of the first visible signs of these interactions are on the plant side the formation of, in the beginning, small bumps, as they are called, on the root system, and later then these bumps develop into a small little specialized organ called nodule, and in these nodules the bacteria finally get established in individual plant cells. The process of the development of these root nodules starts with the attachment of the bacteria on the root surface, and later then these bacteria get internalized into the plant tissue inside of a kind of a small tubing that guides them to the deeper cell layers of the root system. And we learned from our work that apparently at this stage there is quite some stress put on the bacteria. At this moment we do not know exactly what kind of stress this is, but we know that bacteria need the ability to establish these strategies against these stresses or to cope with these stresses. And this is one of the ongoing research activities. And another activity we have in the group is that we like to understand at this level how the two systems communicate and control each other.
Which would, of course, be crucial if you want to engineer it for non-leg use.
Exactly.
As a kind of summary, what do you think is the main outlook, the main benefit for society of your research?
It's a very difficult question. I think what we can contribute is the gain of knowledge, that we can contribute to a better understanding of the interactions between bacteria and plants, I think.
Professor Fischer, thank you very much.
Thank you.
It was a pleasure.